Look at your hand. Every cell in your body carries DNA from species that no longer exist. You're part Neanderthal, maybe part Denisovan. And if you're West African, you carry genetic shadows from a human species we've never even found bones for. Just 15 years ago, scientists thought human evolution was straightforward, one species replacing another in a neat line leading to us. Then they started sequencing DNA from ancient bones. What they found was so strange, so unexpected, that it's forced us to completely reimagine what it means to be human. This is about discovering that Earth was once crowded with different kinds of humans, most of whom vanished so completely, we're only now learning they existed at all. West Africans carry a genetic secret that shouldn't exist. When scientists analyzed DNA from groups like the Yoruba and Mende in 2020, they found something impossible. Between 2 and 19% of their genomes comes from an unknown archaic human, not Neanderthal, not Denisovan, something else entirely. This ghost ancestor diverged from our lineage somewhere between 360,000 and a million years ago. To put that in perspective, that's before Neanderthals even evolved. This mysterious human lived in Africa, bred with the ancestors of modern West Africans, then disappeared without leaving a single fossil we've found. Right now, millions of people walk around with substantial chunks of their DNA, up to a fifth of their genetic code, from a human species we know nothing about, except that it existed. No bones, no teeth, no tools we can definitively link to them. Just genetic shadows hiding in living people. The technical feat of discovering this is remarkable. Scientists use statistical models to identify segments of DNA that look too different from the rest of the genome, segments that must have come from outside the main human lineage. When they spot these anomalies clustered in certain populations but absent in others, they can work backward to figure out when and where the interbreeding happened. Scientists emphasize this explicitly. There must have been archaic humans in Africa about whom we know absolutely nothing beyond these genetic echoes. Every person of West African descent is a living archive of a species that might otherwise have been lost to time completely. Their DNA preserves the only evidence we have that these humans ever existed. 6,000 years ago, hunter-gatherers lived on Colombia's Bogotá Altiplano, 2,600 meters above sea level. In 2025, when scientists sequenced their genomes, they expected to find ancestors of modern South Americans. Instead, they found something that rewrote the textbook, a completely unknown basal lineage of ancient Americans that left zero descendants. Genetically, they represented an entirely separate branch of the human settlement of the Americas. They weren't derived from the North American migrants who were thought to be ancestors of all South Americans. They were something else, a parallel lineage that somehow reached Colombia through a route or at a time we don't understand. For thousands of years, these people thrived in the high mountains. They hunted, gathered, raised families, developed their own culture. Archaeological evidence shows they were successful, well adapted to their environment. Then, around 2000 BCE, they vanished. They did not decline, they vanished, completely replaced by other populations. This discovery shattered assumptions about how the Americas were populated. Scientists, in their own words, didn't believe there was any other lineage in South America beyond the known migration patterns. The idea that an entire branch of humanity could colonize part of the continent and then disappear completely wasn't part of any model. Neanderthals went extinct 40,000 years ago, but their DNA lives on in billions of people. Denisovans disappeared even earlier, yet Tibetans still use their genes to breathe at altitude. But these Colombian Highlanders, who lived into relatively recent prehistory, left nothing, not a single genetic trace in any living human. The mechanism of their disappearance remains mysterious. There's no evidence of genocide, no signs of conflict in the archaeological record. Disease is possible, but wouldn't usually eliminate a population so thoroughly that no genetic trace remains. Absorption through interbreeding is ruled out by the genetic data. If they'd mixed with incoming populations, we'd see their DNA signatures, however diluted. 90,000 years ago, in a cave in Siberia's Altai Mountains, a girl died. She was probably around 13 years old. For tens of thousands of years, her remains lay buried until scientists found a single fragment, a tiny piece of arm bone smaller than your fingernail. When they sequenced its DNA in 2018, they discovered something that shouldn't have been possible. Her mother was a Neanderthal, and her father was a Denisovan. Scientists nicknamed her Denny. She's the only known first-generation hybrid between these two extinct human species. She wasn't someone with distant Neanderthal and Denisovan ancestry. She was the direct offspring of parents from two different species. The chances of finding the one in a million first-generation hybrid should have been essentially zero. Yet there she was, in the first cave where scientists had identified Denisovans just 8 years earlier. The improbability of this discovery forced a radical conclusion. Neanderthals and Denisovans must have interbred regularly whenever they met. Denny's genome tells us exactly how her parents' DNA combined. From her Neanderthal mother, she inherited mitochondrial DNA that matches Neanderthals from Croatia and Germany. Her mother's lineage had traveled thousands of miles across Ice Age Eurasia. From her Denisovan father, she got DNA matching other Denisovan samples from the same cave, suggesting his lineage was local to the Altai region. But Denny represents something bigger than one hybrid child. She proves that the boundaries between human species were far more fluid than anyone imagined. These weren't rare accidental encounters. When different human species met, they recognized each other as potential mates. They formed relationships. They had children together. The cave where Denny lived tells its own story. Denisova Cave was a crossroads where different human species met repeatedly over hundreds of thousands of years. The same site has yielded Denisovan fossils, Neanderthal fossils, and modern human artifacts. It's one of the only places on Earth with direct evidence of all three groups, and the groups there coexisted and interbred. Reconstructions of Denny help us visualize this child of two species. Artists and scientists work together to imagine her appearance based on what we know about Neanderthal and Denisovan anatomy. The result is hauntingly human. A young girl who lived and died 90,000 years ago, carrying the mixed heritage of two extinct branches of humanity. In 2010, scientists sequenced DNA from a finger bone found in Denisova Cave and discovered an entirely new kind of human. They had no skull, no skeleton, just a child's pinky bone and a couple of teeth. Yet from that fragment, they reconstructed the genome of the Denisovans, a species that would turn out to be one of the most successful and mysterious humans that ever lived. Every new Denisovan discovery expands their range dramatically. That first finger bone came from southern Siberia. Then scientists found a 160,000-year-old jawbone on the Tibetan Plateau, 3,280 meters above sea level. Protein analysis confirmed it was Denisovan. These humans had conquered the roof of the world over 100,000 years before our species reached those altitudes. The Xiahe jaw from Tibet revealed something extraordinary. Denisovans visited high altitudes and adapted to them. Living at that elevation requires specialized physiology to cope with low oxygen. Denisovans evolved those adaptations and later passed them to modern humans. Today's Tibetans breathe easily at altitude thanks to a gene variant called EPAS1 that they inherited from Denisovans. Then came more surprises. A jawbone dredged from the seafloor near Taiwan showed Denisovan features. Teeth from southern Siberia matched the Denisovan genetic profile. Each discovery placed Denisovans in a new environment: tropical islands, frozen steppes, mountain peaks. They weren't confined to one region or climate. They were everywhere across Asia during the Middle Pleistocene. The latest revelation came in June 2025. The famous Dragon Man skull from Harbin, China, a nearly complete cranium about 146,000 years old, turns out to likely be Denisovan. Scientists extracted proteins from the skull that carry molecular markers matching other Denisovan fossils. They even recovered mitochondrial DNA from dental plaque that aligns closely with known Denisovan sequences. If confirmed, the Harbin skull becomes the first complete Denisovan skull ever found. It shows a massive brain case, huge brow ridges, and a wide face. A human that would have looked distinctly different from us, but was clearly intelligent and capable. This wasn't some primitive side branch. This was a sophisticated human species that thrived across an entire continent. The genetic legacy of Denisovans reveals their true reach. People from Papua New Guinea carry up to 6% Denisovan DNA. Australians and Melanesians have nearly as much. Smaller percentages appear in East Asians, Native Americans, and even some South Asian populations. The distribution suggests multiple interbreeding events across Asia and Oceania over tens of thousands of years. Hidden in the DNA of modern Mexicans lies one of the strangest stories in human genetics. About 1 in 3 Mexicans carries a gene variant called MUC19 that helps fight respiratory infections. The gene itself isn't unusual. What's bizarre is how it got there. The DNA sequence is Denisovan, embedded within Neanderthal flanking sequences, with a Denisovan core and Neanderthal sequences on either side. This is the first known example of a gene taking such a complex journey between species. The original MUC19 variant evolved in Denisovans somewhere in Asia. Then, probably around 100,000 years ago, Denisovans and Neanderthals interbred, and the gene passed into Neanderthal populations. The Neanderthals carried it west across Eurasia. Finally, when modern humans met Neanderthals, probably in the Middle East or Europe, the gene jumped again, this time into our ancestors. But the gene didn't travel alone. When it integrated into Neanderthal chromosomes, it brought surrounding Denisovan DNA with it. Over thousands of years of recombination in Neanderthal populations, that Denisovan DNA got progressively replaced by Neanderthal sequences. By the time modern humans inherited it, only the core gene remained Denisovan, surrounded by Neanderthal DNA that had replaced the original flanking sequences. Scientists can reconstruct this history from sequence patterns and recombination boundaries. The inner Denisovan core is unmistakable. It matches Denisovan genomes from Siberia. The surrounding Neanderthal sequences match European Neanderthal samples. The boundaries where Denisovan DNA meets Neanderthal DNA show the classic signatures of recombination over many generations. Every detail of the gene's journey is preserved in its structure. The functional importance of MUC19 explains why it survived this complex journey. The gene produces mucins, proteins that form protective barriers in our respiratory and digestive systems. The Denisovan variant appears to enhance immunity against certain pathogens. When the ancestors of Native Americans crossed the Bering Land Bridge into the Americas, they carried this gene with them. It likely helped them survive new diseases in their new world. This isn't the only gene with such a twisted history, just the clearest example we've found so far. Geneticists are discovering more instances of what they call adaptive introgression, useful genes jumping between species through interbreeding. Some immune system genes in Europeans show signatures of passing through multiple archaic populations. Certain metabolic genes in Asians may have taken similarly complex routes. Every person without recent African ancestry carries between 2 and 3% Neanderthal DNA. People from Papua New Guinea and Australia carry up to 6% Denisovan DNA. These aren't random genetic artifacts. They're functional genes that still shape how our bodies work today. We're walking around with borrowed adaptations from extinct humans, genetic gifts that help us survive in specific environments. The most famous example is the EPAS1 gene in Tibetans. This Denisovan variant allows them to thrive at altitudes where most humans struggle to breathe. At 4,000 meters above sea level, where oxygen levels are 40% lower than at sea level, most people develop altitude sickness. Their bodies produce too many red blood cells, thickening the blood and straining the heart. Tibetans with the Denisovan EPAS1 variant don't have this problem. Their blood remains normal even at extreme altitudes. Neanderthal contributions are more varied but equally important. Multiple studies have identified Neanderthal genes affecting skin and hair in European and Asian populations. One variant influences skin tone, helping populations adapt to lower UV radiation at high latitudes. Another affects hair texture and thickness. These aren't cosmetic changes. They're adaptations to cold climates that Neanderthals perfected over hundreds of thousands of years in Ice Age Europe. The immune system shows the strongest signature of archaic admixture. Neanderthal and Denisovan genes are overrepresented in immune-related regions of our genome. The genes that help us fight viruses, bacteria, and other pathogens often trace back to our extinct relatives. When modern humans expanded out of Africa, they encountered diseases their immune systems had never seen. Archaic humans had been dealing with these Eurasian pathogens for hundreds of thousands of years. Through interbreeding, modern humans instantly acquired tested immune defenses. Some of these immune genes have unexpected modern consequences. Certain Neanderthal variants that boost immune response are now associated with allergies and autoimmune conditions. Genes that helped our ancestors survive new pathogens can overreact in modern environments, causing our immune systems to attack harmless substances or our own tissues. The same Neanderthal DNA that helped our ancestors survive might contribute to hay fever today. Metabolism genes tell another story of archaic adaptation. Several Neanderthal variants affect how we process fats and carbohydrates. One influences lipid metabolism in ways that would have been advantageous during feast or famine cycles of Ice Age hunting. Another affects insulin sensitivity. In prehistoric environments, these genes helped our ancestors survive food scarcity. In modern contexts with abundant calories, some increase risk for diabetes or obesity. The distribution patterns reveal the history of human migrations and adaptations. East Asians carry different Neanderthal genes than Europeans, indicating separate interbreeding events with different Neanderthal populations. Some Denisovan genes appear only in Oceanians, others only in East Asians, suggesting multiple Denisovan populations adapted to different environments. Even brain function shows archaic influence. Some Neanderthal variants affect neurotransmitter synthesis and neural development. While we can't directly measure their cognitive effects, their persistence suggests they weren't detrimental and may have provided advantages we don't yet understand. What's remarkable is how precisely targeted some of these archaic genes are. The Denisovan altitude gene helps at high elevation, but provides no advantage at sea level. Neanderthal skin genes help at high latitudes, but aren't beneficial near the equator. Immune genes that fight specific pathogens only help where those pathogens exist. June 2025, scientists confirmed that the Dragon Man skull from Harbin belongs to a Denisovan. This wasn't a small revision. It completely changed our picture of what Denisovans looked like. For 15 years, we'd imagined them based on a finger bone and a few teeth. Now we have a face, massive brow ridges, a wide skull, a brain as large as ours. The fossil had been known since the 1930s, sitting in collections, studied repeatedly. Only new protein and DNA extraction techniques revealed its true identity. This keeps happening. Fossils we thought we understood turn out to be something else entirely. Genetic analysis repeatedly overturns decades of anatomical study. Each technological advance, ancient proteins, improved DNA extraction, statistical modeling of genomes, reveals another layer of human prehistory we didn't know existed. Consider what we've discovered just since 2010. An entire new human species identified from a pinky bone, a ghost species in Africa known only from genetic shadows, a first-generation hybrid of two extinct species, a vanished South American lineage that left no descendants, genes that hopscotched between species, altitude adaptations inherited from extinct relatives. Each discovery was completely unexpected. The pace is accelerating. New techniques extract DNA from cave sediments where no bones survive. Protein analysis identifies species from fossils too old for DNA preservation. Statistical methods detect ghost lineages from patterns in living genomes. Artificial intelligence models predict ancient population structures from genetic data. Every method reveals human populations we didn't know existed. We're also discovering the absences, the gaps that suggest missing populations. Huge regions of Africa and Asia have no ancient DNA samples. Entire time periods are blank. We have Denisovan DNA from 200,000 to 30,000 years ago, but almost no fossils. We have Neanderthal fossils across Europe and Western Asia, but little from their earliest periods. Each gap might hide unknown species, ghost lineages, vanished populations. Interbreeding patterns suggest even more complexity. If Neanderthals and Denisovans produced fertile offspring whenever they met, how many other combinations existed? Did the African ghost species interbreed with other archaic Africans? Were there hybrid populations that themselves became distinct groups? We only detect interbreeding when descendant populations survive to the present. Failed hybrid lineages remain invisible. The molecular revolution in archaeology has just begun. Most ancient fossils haven't been tested with modern techniques. Museum collections worldwide contain potential Denisovans, ghost species, unknown lineages. Every year brings better extraction methods, more sophisticated analysis, larger reference databases. Fossils that yielded no DNA a decade ago might be sequenceable tomorrow. Living populations hold more secrets. We've thoroughly studied European and East Asian genomes, but many populations remain unsampled. Indigenous groups in remote regions might carry signatures of unknown archaic humans. Island populations might preserve unique ghost lineages. Each genome sequenced might reveal another extinct human species through the DNA it left behind. Some scientists estimate we've discovered less than half the human species that existed during the Pleistocene. Others think we're missing entire categories of human diversity. The genetic evidence suggests a world where different human species were the norm, not the exception, where interbreeding was common, where populations appeared and vanished, leaving only genetic shadows or nothing at all.